So no news today. You have to read the paper yourselves. Okay, and so I will introduce our speaker. We are so pleased to have Trevor Bedford joining us today. He is at the Fred Hutch Cancer Center, as well as the University of Washington. He is really a visionary epidemiologist, modeler, and really kind of pushing the envelope in the use of sequence data from viruses to understand and predict how they emerge, how they spread, how they evolve as they circulate in our communities and around the globe. And not only does his work help us to understand these processes and give us strategies for combating viruses, but his work is literally visionary in that his evolutionary trees have indelibly impacted how most of us think about and conceptualize epidemics. And in February, Dr. Bedford's done a lot of great work, but in February, I think really noteworthy, he sort of connected the dots for us all between the early COVID arrivals from Wuhan and subsequent community cases in Seattle, making it clear that COVID was here and it was spreading silently, and we had a big challenge on our hands. And finally, I would be remiss if I did not mention Dr. Bedford's Twitter stream, through which he fearlessly helps us all as researchers and the public to navigate the unfolding COVID situation and the really hard science. So thank you so much for being here, Dr. Bedford, and please take it away.
Oh, thank you, Lauren. That was a very very wonderful introduction. It's been very strange having kind of one of the more important things that I'm doing be Twitter, but still I think it is important. So I wanted to just briefly go over the kind of genomic side of SARS-CoV-2, and then kind of look at both from epidemiological and evolutionary perspective. So I'm assuming everyone can see my screen, and I should say that slides are up at bedford.io/talks if you want to get any of these slides. So we're all used to looking at things like this. I look at these every day, and I'm sure everyone in this room does, and in various kind of geographies. Case counts are extremely helpful, and then it's also helpful if we have some kind of modeling layer on top of that to be able to help make sense of these case counts. And so often we might be interested in terms of a forecasting challenge of will they go up or will they go down, et cetera. But what I've mainly focused on is that generally we have this kind of significant fog of war. We don't really know what's going on. It's hard to know whether these case counts are a 5x underreporting or maybe they're 20x underreporting. We can kind of combine that with serology, and you can do other work to figure this out, but it is still a challenge. And so generally, I believe that genomic approaches offer an interesting orthogonal data source to understand the pandemic. They're not meant to replace case counts or serology, but kind of gives an alternative perspective on things. And this is kind of the toy example that I often use, where I'm assuming people can see my cursor, where there's an infection here with this red dot, and then that spawns off a couple of secondary infections, which cause their own secondary infections and so forth. And what you can do is you can sample some of these individuals, and then take a nasal swab, sequence the infecting genome. And then what you'll find is patterns of shared variation. So we'll have, say, this purple mutation here that these two infections share, this orange mutation that these 4 infections share, and this blue mutation that just this one has. And we can take this pattern of shared variation to be able to reconstruct a phylogenetic tree. And so then it becomes a challenge in both, mainly in interpretation, to be able to connect to this tree and then understand something about this underlying process. And you can either do that by interpretation or by kind of statistical models, phylogenetic models that are layered on top of this tree. So we've been trying to make this genomic epidemiology actionable for a while now. This is with this Next Strain project. And the idea was basically to do the things that show up in every paper that you read on genomic epidemiology, but have this kind of happen just in a very routine pipeline. This is a project with co-founded by Richard Neer, and then James, Emma, Tom, Louise, et cetera, have really led things for the last while. The general architecture, I should say that everything is completely open source up on GitHub /NextStrain. It's been pretty amazing during the pandemic, having so many kind of open source contributors. There are now just everyone's kind of filing issues and filing pull requests and contributing to the code base, which is really cool. The general setup is that we have our own in-house database that we're actually not using for SARS-CoV-2, but we've used for other things. We have a Augur, which is kind of Python processing scripts to be able to go from sequence data to an alignment, to a tree, to a time tree, to a geographic inference tree. And then that goes then to Auspice, which is our front-end web visualization in JavaScript, and it's the kind of the thing that you actually interact with. For right now, we're up to, I think, 25,000 SARS-CoV-2 genomes, which is amazing. But that is now this significant challenge in terms of processing. And so what we generally do when we have too much data, and this is generally what the field does as well, which is a little unfortunate, is that we have to significantly subsample. This is necessary for computational reasons, but it's also helpful in terms of representation, where it's good for these models to try to subsample so that you're getting roughly sampling proportional to prevalence, if you can do that. And so the 30,000, 25,000 SARS-CoV-2 genomes are really heterogeneously sampled across the world. About half of them come from the UK right now, and a significant portion from the US. And then in the US, it's like 2 different states that are contributing most of the genomes. And so, so we subsample down, then we can align with MAFT. That takes about 20 minutes. We generally aim for 3,000 or 5,000. We build a maximum likelihood tree with IQ tree, and we temporally resolve this tree and geographic ancestry with tree time. This whole thing takes about 2 hours. So the current data flow is, I should have updated this, that we have everything goes into GISAID. GISAID has been helpful this way because they do a lot of curation, and they've made it very easy for submitters to upload genomes, which is great. It's a little, so genomes get uploaded to GISAID, and then we can be able to pull down a complete data set from GISAID every 60 minutes as kind of just an automatic thing that's running. This will trigger a rebuild on AWS. We look at things, make sure that we don't have to add new lat longs or something like this. And then we push this online to nextstrain.org and tweet an update. Right now, we're doing this about every 12 hours. We have a Seattle shift and a Basel shift to look at the new data. We were regularly getting about 200,000 visitors per day, and now we're down to about 50,000 as interest has flagged a little bit. If we look at the data, we can see that early on, if we go from December 15th to January 15th of samples, basically everything is still in Wuhan at that point. And one of the first things that we see, and this is kind of one of the early observations from the genomic epi, is that all of these samples from Wuhan are very closely related genetically. They differ by maybe 1 or 2 mutations at the most. And that basically means that we have to have a recent common ancestor to that genetic diversity in Wuhan that places it at late November or even early December. And so the spark that eventually caught in terms of human to human transmission was this one event that happened late November, early December, and then spread from there, even if there were other zoonotic cases going on in the seafood market. So then if we go from January 15th to February 15th, we see that then we get this large epidemic within China, and which are these blue dots that you see here. And then we can see these other dots that are viruses sampled from elsewhere in the world, Europe in kind of yellow-green, US in red. And you can see that in that case, the things that we're catching between January 15th and February 15th are really these one-off introductions, these sparks that land that were contained, and they don't really catch on. However, during that time, there were other sparks landing that were just not detected by surveillance apparatus in Europe or in the US. And so that's kind of, you can see these branches over here and this branch over here and so forth. And so if we go forward from February 15th to March 15th, we can see that then there's this huge amount of cases of infections that derive from these arrival events that occurred in January, February that just weren't detected. And then we kind of go forward from there, from March 15th to April 15th, where now we kind of have this evolution, this spread that's happening within regions, and not really mixing quite as well. And so we kind of this initial kind of event where everything gets distributed through the world, and then more local transmission after that point. If we look in the US in particular, so we can see that this is kind of just zooming in. Each red dot is a infection in the US and showing how they're related. We have this large clade here down near the bottom of the tree that appears to be a direct introduction, a single introduction in late January directly from China, that then spread for this kind of all of February before being detected, basically. And then we also have other events where the kind of European epidemic is taking off at this point. And then we have multiple introductions from Europe into the US, into New York, that then kind of fueled things. If we look within the US, now this is kind of same idea. Each dot is a sample from the US. Orange are from the West Coast, green are from the East Coast. And what you can see is that there is a lot of mixing, so that once it arrived here, it kind of spread rapidly through the country during February when people weren't really thinking about things. If we look, and we can kind of see that even more, let's say if we look at this clade down here and zoom in on it, again, we have this arrival at the beginning of February, and then it shows up really strongly in Seattle and Washington, but you can quickly find it elsewhere, in British Columbia, in New York, in Texas, where kind of even in the span of just this month, it got to a large number of places. So in the absence of restrictions, there's a lot of kind of mobility to the virus. So generally, I think that the sequencing is very useful for epidemiological understanding. And what we're trying to do now is get groups like state health departments set up so they can kind of have a view of Wisconsin, a view of Texas, a view of whatever kind of geography you're interested in, to be able to kind of make more localized inferences, because the data set's kind of getting too big for us to do much on the besides a global or continental kind of views. But in addition to this epidemiological understanding, I think that selection and functional impacts are interesting. And I wanted to discuss this recent Korber et al. Bioarchive paper, because it's gotten a lot of attention and I think important to discuss. So in this paper, they looked at this mutation in spike, so which binds to human ACE2 and is a very important protein for the virus. This mutation that goes from D to G at site 614, highlighted here. And so what Korber et al., and so if you look at this mutation, we see that that interestingly, it basically appeared almost completely coincident to this European introduction. So this is kind of the same picture we're looking at before. Purple lines are in China. D614G appears once in the virus population here with this yellow star. And then basically almost immediately after that, a transmission chain that has that mutation is the thing that arrives into Europe in this green branch here, and then kind of spreads from there. And so there was a this is obvious happenstance, where the virus that happened to land in Europe was the virus that had D614G. And so we have this confounding here that will make it difficult to fully take apart evolution epidemiology. So if we look then at D614G, because of this attachment to this branch here, what we see is that D is prevalent in China, Southeast Asia, much of East Asia. Whereas if you look at Europe, it's almost completely G, because it was founded by this arrival event. Whereas the US, which was seeded by both Asia and Europe, has a mix of the two variants within it. And same with Australia, which was seeded by the US and by Europe primarily, and by Asia as well. And so if we look, however, and so we see this mix, however, if we look at individual regions, we see this interesting pattern where within regions we get an uptick of G over time. And so basically, California has become largely G, Minnesota kind of completely came to dominate, regions in Australia increased as well. And basically every region that we've looked at, we get an increase in G over the D variant. And this is kind of showing the same thing, that over basically 6 weeks, we get a substantial increase in G. And this can be due to either that the 614G could be more transmissible. It's definitely possible. And that it has a higher R naught through some mechanism that we don't really understand. And so that it's outcompeting, and that transmission chains that have the G variant tend to be larger than the D variant. And so over time, that has come to dominate the population. Or everything could be these founder effects, and that there was a lot of G in Europe that kind of repeatedly seeded outbreaks elsewhere, and that has come to dominate that way. So picking apart these two things is quite difficult. One thing that we were able to look at, however, so this is from Corber et al., where in Sheffield, England, they see that in a cohort of patients, that individuals with a G mutation tend to have lower CT values, which means higher viral loads. So that kind of maybe indicates a mechanism by which you could have more transmissibility. We were able to repeat the same analysis for samples for individuals from Washington State, and then also see that G has a lower CT value or higher viral load. So that's maybe kind of another data source that suggests that this might be a thing. So generally, moving forward, I think that this genomic epi has a large potential for surveillance. Again, we're trying to get state labs, countries set up with kind of instances of NextStrain to run things themselves. We'll have in the coming years, based upon what we've seen for seasonal coronaviruses, I think that we're going to need to update the vaccine and to have a good idea of what's circulating for this. And so kind of this evolutionary tracking will be and forecasting will be very important for that. And then generally, as I think everyone agrees, that right now the best way out of this mess is with test, trace, isolate, and to kind of really ramp up that capacity. And I'll stop there and take questions.